Mitosis is the process of how cells divide and replicate themselves for growth or repair, during which the nucleus of a single cell will split itself into two daughter cells, creating two sets of the genetic material. Cells that have a nucleus which contains its genetic information are called eukaryotic cells. Animals, plants, and fungi are the main examples of eukaryotic organisms. The nucleus of a cell contains chromosomes. Chromosomes are thread-like structures found within the nucleus of most living cells. Each chromosome is made of DNA molecules and carries a large number of genes. A cell spends most of its life in a state in interphase, which takes place before mitosis can actually begin. Interphase has 3 stages. The first is called G1 phase, during which the cell grows larger and carries out different functions depending on the type of cell. Once the cell has grown too big to function, S phase or synthesis begins. This is where the cell makes a copy of all its DNA. The last stage of interphase is G2, where the cell grows more, makes proteins and organelles, and prepares itself for mitosis. Now that interphase is complete, the 4 stages of mitosis can begin. As you can see at this point in the cell cycle, the DNA within the nucleus is all in the form of loose chromatin, which looks sort of like spaghetti. During the first stage of mitosis called prophase, the loose chromatin condenses into X-shaped structures called sister chromatids, which are the 2 identical copies of a single chromosome created during S phase. These are connected at the center by something called a centromere. Next, the nucleus of the cell disassembles in order to give spindle fibers access to the chromosomes. Spindle fibers are tubes made of protein that are used to move chromosomes around. They attach to the chromosomes through the centromere. Now we have arrived at metaphase. During this phase, the spindle fibers will move and reorganize the chromosomes neatly down the exact middle of the cell, so that a sister chromatid is on either side of the cell. Next phase is anaphase, where the sister chromatids are separated from one another and pulled to the opposite side of the cell, each one forming a V-like shape. And lastly, we have telophase. Before we can create 2 new totally separate cells, we must dismantle the spindle fibers, uncoil the chromosomes back into loose chromatin, and create a nuclear envelope around each set of chromosomes, creating 2 new nuclei, essentially resetting everything back to how it was during interphase. Except this time we now have 2 identical cells. Finally, a process known as cytokinesis is used to fully separate the cells from each other.